Dr. Walsh is a postdoc researcher whose dissertation focused on the development of molecular pathology techniques to better understand wild fish health and disease. She has worked at the Eastern Ecological Science Center in Kearneysville, West Virginia for 15 years under the supervision of Dr. Vicki Blazer. Her current research deals with looking at the effects of contaminants associated with industry, agriculture and best management practices, and PFAS on the health of wild fish. So Heather, we'll turn it over to you.
Okay. All right. So to wrap it up here, I'm going to give you a talk on the study we've done on the reproductive health and endocrine disruption we identified in smallmouth bass from the Lake Erie drainage. To give you a little bit of background, in 1987, the Presque Isle Bay was listed as an area of concern due to fish tumors and other deformities, the beneficial use impairment, and also because of restrictions to dredging because of contaminated sediments. The sources of contaminants included industry and still continues today, primarily from PAHs, PCBs, and other legacy contaminants. And some newer emerging contaminants are also coming from wastewater in this area. I have a map here of Presque Isle Bay, and you can just see how developed the land use is around it. And so pharmaceuticals can also become an issue in this area. In 2012, however, the area of concern was delisted, but this was somewhat controversial because some of our work that we did in this area showed that the fish tumors didn't necessarily drop below the levels that warranted delisting. So there was some public outcry on the matter. A little background on smallmouth bass in the Lake Erie drainage. They are a native species, and they're one of the top 4 species that are targeted by anglers in this area. And they are a highly sought after sport fish in most of the United States. There are not health concerns currently on smallmouth bass in the Great Lakes as what we've seen in Potomac and the Susquehanna River drainages. But previous sampling that we've done here, we found endocrine disruption. We found vitellogenin and intersex in male smallmouth and largemouth bass. So part of our biological effects-based monitoring has included to investigate the potential reproductive endocrine disruption in the Presque Isle Bay, specifically Misery Bay and 2 nearby tributaries. We included histopathology, transcriptomics of the testes and the liver transcriptome was published by Hahn et al. In 2016. Let's see, gene transcript abundance, we analyzed from the liver and the testes as well, and we targeted reproductive immune function and contaminant metabolism transcripts. As part of this monitoring, so in the spring, May 2016, we sampled about 30 adult smallmouth bass from Elk Creek, 20 Mile Creek here, and Misery Bay. We weighed, measured, and bled the fish, and we bled the fish in order to extract the plasma for vitellogenin analysis. And we weighed the testes, we weighed the gonads, because we also sampled females, and the liver in order to calculate the organosomatic indices. We took pieces of the gonad and liver in Z-Fix for histopathology and small pieces in RNA-later to do the gene expression and transcriptomics work, and otoliths for aging. Some land use analysis was also conducted using the 2016 land cover data from MRLC. The catchment size was calculated as well as the % land cover at each site. So as for part of the histopathology, we analyzed 5 to 8 cross sections of the gonad, and TO severity was also analyzed, which was calculated from Blazer et al. 2007's TO severity index. So here's pictures of what the severity rankings would look like. A TO severity of 1 would be just like one single oocyte. And then as you get to 2, you see an increase in the number of oocytes, but there still aren't very many and they're fairly spread apart. With 3, you see an increase in the number of oocytes and you see more clustering in a single area. And then 4, you just see quite an increase in the number and clustering within an area of testicular oocytes. Because we didn't have control male fish that we could confirm 100% were not intersex, we couldn't actually do differential expression analysis comparing normal and intersex fish. So instead, we decided to look at comparing testicular oocyte severity to female fish to see how they would compare. If, as the severity increases, how does that feminization compare to the females? So with our TO severity rankings, we rank them low, medium, and high with our scores, the mean scores. So low was 0 to 0.5, a medium intersex fish was 0.6 to 1.5, and high was greater than 1.6. And I can say that in this study, overall, the severity was low, and we didn't see any fish with TO severities of 3 or 4. So they were within 1 to 2. And this is how we did our differential gene expression comparing to females. We analyzed 3 cross sections of the liver, and also the plasma vitellogenin was analyzed with a direct ELISA assay. The liver transcriptome was published by Hahn et al. In 2016, as I previously mentioned, and here's the citation for it if you're interested. But for the testes, that's not been published yet, but will be in this paper. I pooled individuals for this analysis. I pooled multiple intersex males for one sample, a group of non-intersex males for the next sample, and then I also included immature females, thinking that this would help to include genes expressed by females. And I targeted immature females because if you noticed in the picture of the testicular oocytes, the oocytes never, in smallmouth bass at least, they never advanced beyond the primary oocyte stage, which is what we see in immature females. And these samples were sequenced. Oh, and I also want to say that these were normalized to the lowest concentration within each pooled sample. And the sequencing was done at the Institute for Genome Sciences in Baltimore, Maryland on the HiSeq 4000. So for gene quantification, I use the nanostring N counter. If you're not familiar, I'm not going to get too in the weeds with how the technology works. It takes up a little bit of time, but if you have any questions about it, let me know. But I can say that the N counter, the data that you receive back is similar to what you would get with qPCR. That's quantification and count data. So Hahn et al. Also used this analysis, and they developed a suite of about 50 transcripts that they analyzed in the liver. For this paper, though, I narrowed it down to transcripts that were more associated with reproduction. So I think we focused on, it was either 17 or 19 out of those 50 transcripts. For the testes, though, in order to select the transcripts that I wanted to analyze in the code set, I looked at which transcripts were differentially expressed between the samples that were used in the transcriptome. And I also looked at what transcripts had been found to be associated with intersex from the literature. So I ended up with a code set that consisted of about 40 transcripts that were associated with reproduction, immune function, and development. Statistical analysis I did in R, not listed here, but I did a Dunn's test to look at site differences for these biological variables, and then the Spearman's correlation to look at associations between the biological variables and transcript abundance. For the differential expression between the low, medium, and high TO severity males compared to females, I did that in an open source software called NanoString Diff. I had a false discovery rate cutoff of 0.05 and the log fold change of plus or minus 1.5. Sometimes some studies use 2. to be more conservative, but that kind of would have taken out some of the significant findings I found. So here's some results in the land use. Elk Creek definitely had the largest watershed area, but Misery Bay won with the most developed land use. Quite a bit different. If you recall seeing that picture I showed earlier of Presque Isle Bay, Misery Bay, across from all that development is what we consider Misery Bay, but it still had very high rate of development compared to Elk Creek and 20 Mile Creek. 20 Mile Creek actually had the largest forested area, and Elk Creek had the highest amount of agriculture, much more so than Misery Bay, but somewhat more than 20 Mile Creek. And Elk Creek also had the highest percentage of cultivated crops, which can be of concern when you're thinking about the difference between like a pasture hay landscape versus cultivated crops. Because oftentimes in a pasture hay type of agricultural setting, they're not putting pesticides and herbicides on the land, but they are for cultivated crops. So that can be something to keep in mind. As I mentioned, we also sampled females, but I didn't include them here for sake of time. So here, from here on out, I'm going to present just the male data. At Misery Bay, and remember, we collected 30 fish at each site, but at Misery Bay, we only collected 4 females. So we had quite a few males that we analyzed here. We like to try and get a 50-50 ratio if that's possible. Misery Bay had the largest fish, but 20 Mile Creek had the oldest fish. So there could be something going on here where prey might not have been as abundant at 20 Mile Creek. Maybe so it's a foraging effect, or maybe even temperature difference. If Misery Bay was warmer than 20 Mile Creek, the fish could grow larger faster. So although 20 Mile Creek had the largest forested area, it had the highest amount of plasma vitellogenin found in both the males and the females. And in fact, it was significantly greater. I can't see my mouse. It was significantly greater than at Elk Creek and Misery Bay. So I forgot to mention the lowercase letters indicate significant differences. And then intersex severity was highest at Elk Creek, which had the greatest amount of forested landscape, which was in the form of mostly cultivated crops, and it had the highest levels of intersex in the males. Although it had the highest numbers of intersex males, if you look across the board here, Misery Bay and 20 Mile Creek also had a fairly high prevalence of intersex. So to jump into the results of the transcript correlation to the biological variables. So in the testes, the gonatosomatic index was negatively associated with follistatin 3. And this was kind of interesting because from what I found in the literature, that mice that were deficient in follistatin 3 actually had enlarged testes because it was associated with the increase in Sertoli cell numbers. They said it didn't have an impact on reproduction, which we generally don't see in smallmouth bass, but it would be interesting to find out whether or not this was the association, and maybe some Sertoli cell counts in the future could tease this out. Intersex severity was positively associated with insulin-like growth factor binding protein 1. So this transcript has been found to be upregulated in intersex rainbow darters and upregulated in response to metformin exposure. And metformin is a diabetic drug, which has been shown to induce intersex in fathead minnows. So lastly, plasma vitellogenin was positively associated with 3-beta hydroxysteroid dehydrogenase, dehydrogenase reductase 11, and follistatin. The 2 hydrogenases are involved in steroid hormone biosynthesis, so that could be that would kind of make sense with plasma vitellogenin. And follistatin promotes, in males, it promotes spermatogenesis, but in females, it promotes oocyte maturation. So that would also kind of jive with being associated with plasma vitellogenin. It's nice when things kind of make sense. In the liver, TO severity was positively associated with I think I'm going to butcher the name of this transcript, but diiodeinase 1. Although the correlation wasn't very high, you know, p-value is just below 0.05. It is kind of interesting because DIO1 is associated with the conversion of thyroid hormone T4 to T3, and T3 has been shown to regulate testes growth and Sertoli and Leydig cell differentiation. So if there's some kind of a disruption in this pathway that might be associated with the differentiation of these cell testicular oocytes, that would be something that would really be cool to study on further. So for plasma vitellogenin, the transcripts that were there's a handful of transcripts that were negatively associated with plasma vitellogenin, and some of these include the androgen receptors that are associated with male maturation, including estrogen receptor beta 1, which you think of estrogen receptors and you think of females, but it's also been found to be downregulated in male zebrafish exposed to EDCs. AHR, aryl hydrocarbon receptor. In female fish, it's oftentimes positively associated with plasma vitellogenin. So to find it negatively associated in males, I'm not really sure why that's occurring, but maybe it's a sex difference and something that also warrants further investigation. And you'll note here that DIO1 is negatively associated with plasma vitellogenin. And in our studies, we often don't find plasma vitellogenin and intersex to be correlated with one another. So this transcript maybe helps explain why we see that discrepancy, and that's also been found in other species of fish. Lastly, there was 4 transcripts positively associated with plasma vitellogenin: CYP3A, estrogen receptor alpha vitellogenin, and choriogenin. The last 3, ERA, VTG, and choriogenin, we would expect to likely see positively associated with plasma vitellogenin. But it kind of cool because I found a study that was done in Medaka where they exposed the male fish to synthetic musks, and these CYP3A, ERA, and VTG were also positively associated with plasma vitellogenin. So that's also kind of an interesting finding. And those correlations are a little bit stronger than some of the other ones. So the results of the differential expression. So obviously this could only be done for the liver, not the testes transcripts, because I did not analyze the ovaries the same way. So there was not a whole lot of significant findings here. These 3 transcripts that were differentially expressed between the low, medium, and high males and females, not really surprising. Again, choriogenin, estrogen receptor alpha, and vitellogenin. Those are oftentimes much higher produced in females, and even though we have feminized males that we're comparing them to, the levels were still much higher. Androgen receptor alpha, I would have expected this finding to be the opposite. I would have expected to see a significant difference between the high TO severity males and females, mostly because you would expect an androgen receptor alpha to be maybe depressed in the more feminized males than the lower feminized males. So that finding, I'm not really sure how to explain. GLK, glucokinase, this could be reflective of the increase in glycogen production, where in the in the males with more testicular oocytes, since as oocytes mature, they incorporate more glycogen into the cell. But I want to point out here, there are a lot of transcripts that were not different between the males, the intersex males and females. Whether or not this is a significant finding on its own would need to be answered by having true control males compared to females, because as I pointed out, estrogen receptor beta has been found to be reduced in male fish exposed to EDCs. But, androgen receptor beta is not different, and some of these others. So it would be kind of interesting to find. And maybe we wouldn't expect some of these to be different because they're not directly associated with reproduction, but maybe development or contaminant metabolism. So to conclude, I'm not going to reiterate what I've already said, because I think the conclusions kind of can be wrapped up differently by pointing out that we did find multiple indicators of endocrine disruption, both chronic and acute indicators. So chronic would be intersex. Intersex has been shown to be induced early in life in fish when they're exposed to EDCs as fry or as juveniles. So, and there was quite a high rate of intersex. Although we didn't see high severity of intersex, we have found in previous work where we've sampled at the same site over many years that intersex severity can change, and that's more plastic than intersex prevalence. And then we found acute indicators of endocrine disruption. Vitellogenin is often induced weeks to months after exposure, and transcripts are induced hours to days. But when you're looking at transcript data, you do have to take in some consideration that these fish are moving. And yes, we sampled them in these tributaries, but they're probably moving in and out of Lake Erie itself. So where the exposure source is coming from is not clear, and it's not always going to be clear when you're dealing with wild fish. And that's why, validating with laboratory exposures is really key to maybe validating some of these transcripts that I have talked about as potential biomarkers. But laboratory exposures themselves can have their own caveats, because these fish exposed to all these different types of contaminants in the wild, you can never really recreate that in the lab 100%, and the mixtures of these chemicals and the effects that they might have. So, and lastly, I just want to point out that, I did not present any water chemistry data here because we don't have any. So really, it would have been nice to have water chemistry data to try and associate with what we saw with the transcript data, but such is life, and it's not always perfect. So I want to thank the field crew for helping, process all these fish. We sit out there by the side of the river and work them up. Adam Sperry did the plasma vitellogenin analysis, and the ladies from the histology lab, all of which have since retired, and Adam has taken their place, but at the time they prepared the slides. The funding was provided by the Pennsylvania Sea Grant and the USGS Ecosystems Mission Areas Environmental Health and Fisheries Program. And with that, I will take any questions.
Great, thank you very much, Heather, for an excellent talk. If folks have questions, feel free to unmute yourself, or you can drop them in the chat box and I can read them to Heather.
I accidentally closed out the videos of everybody, so I can't see. It was blocking the slides as I was looking at them. So I don't know how to bring that back up.
You could probably stop sharing now.
Oh, okay.
bring things back for you. But Ben, you have a question, looks like.
Oh yeah, yeah, great, cool
Okay.
talk, Heather. It's really interesting,
Sorry.
and I think really cool application of RNA-seq. I wasn't familiar with this nanostring encounter, and I was just curious what the decision was to choose something like that assay over like a normal like a transcript read counting assay.
So instead of like doing RNA-seq on multiple individuals or even qPCR, the benefit of this is that you can analyze 12 samples at a time and up to 800 transcripts, which obviously we didn't do here because as I pointed out, we did 50 transcripts from the liver and 40 from the testes. So it's definitely, it's somewhat cheaper than RNA-seq. The benefit is that you can directly analyze the transcripts that you want, and there's not a lot of data associated with it. So like it produces kilobytes worth of data instead of like gigabytes. So it's easier to analyze. You can directly target the transcripts you're interested in a higher number of samples. Because we've analyzed at this point hundreds of samples, which would be crazy to do with RNA-seq.
Cool, that makes sense. No, thank you.
Any other questions out there for Heather? All right, if not, thanks very much, Heather, for the great talk.